Greetings everyone, my name is Kevin Chappell and I'm a PhD student from the University of Arkansas at Little Rock and the University of Arkansas for Medical Sciences. Today I'll be giving a presentation on a paper published by our lab on how multi-omic integration reveals correlated regulatory features of triple-negative breast cancer. The goal of our project was to use multiple omic data types and integrate them using machine learning algorithms to try and uncover significant biological features that may have otherwise been difficult to detect using a single omic framework. For our experiment, we used triple-negative breast cancer data harvested from three separate cell lines, MCF10A, MDA-MB231, and HCC1937. The MCF cell line represents our control as it's made of benign epithelial breast tissue. MDA-MB231 is our experimental cell line that comes from a patient with late-stage triple-negative breast cancer, and HCC is our other experimental cell line that has triple-negative breast cancer but also has a germline mutation within the BRCA1 gene. Each of our cell lines were subjected to multiple independent experiments to properly obtain each omic data from the samples, such as DNA methylation, RNA-seq, protein analysis, and PTM analysis, before it was then integrated into multiple machine learning algorithms to obtain our final results. Before inputting our results into these machine learning algorithms, however, we first tested the correlative strength between each of our different samples and omics datasets using a Pearson's correlation analysis. From these results, we found that both genes and proteins, as well as proteins and phosphorylation, all had very good correlation between one another. However, the DNA methylation and gene expression seem to not correlate, but it can be seen significant as there's a positive beta value in the DNA methylation, which indicates hypermethylation, which actually leads to decreased gene expression. So there's small correlation, but perhaps not as significant as genes and proteins and phosphorylation and proteins. Once we were confident in how each dataset would correlate between one another, we integrated our data into two independent machine learning algorithms. First, we used a tool known as MixOmics to analyze our data on a feature level. MixOmics is a supervised analysis that was used to identify key features from multi-omic data that can clearly separate between the three cell lines we use to create our data. Using this tool, it creates a number of principal components that can separate our data from one another. The first principal component that we viewed separated our HCC1937, so our BRCA1 mutant cell line, from the other two cell lines in the data, and found important features such as the TGF-BETA 1 and TGF-BETA R2, KLF6 and KLF12, PIK3R3, and VIM to be significant. Next, we took our data and integrated it into another tool known as Multiple Omic Gene Set Analysis, or MoGSA. This tool, machine learning tool, uses a low-dimensional representation of the most variant correlated features across the different data types and transforms them to scale. This tool has a greater focus on creating a gene set score, and it's better for obtaining results pertaining to pathways instead of individual feature level like MixOmics. MoGSA then creates a gene influential score, which we can use to identify a number of important pathways within our dataset. One of such pathways we took for further analysis that also was corroborated by our MixOmics results was the TGF-BETA pathway. Interestingly, here we observed that TGF-BETA R1 protein expression and SMAD1 phosphorylation are upregulated in our MDA-MB231, so our normal BRCA1 samples, compared to the MCF10A control samples. This interesting finding raises the possibility to target the TGF-BETA, TGF-BETA R1 SMAD1 through 5 axis to inhibit pro-tumorigenic functions of TGF-BETA signal transduction pathway. These results still need to be validated in lab before any further concrete results can be shown. Putting all of these findings together between both single omic analysis and the results we obtain from both mixed-omics and OGSA, we find a cascade of different features among separate omics levels that work together. In this example below, we demonstrate how hypomethylation of a promoter region and increased levels of histone and proteins can lead to a downregulation of PTEN and beta-catenin. This function shows that BRCA1 enhances the levels of nuclear active form of beta-catenin, whereas the mutant BRCA1, such as our HCC 1937, can actually limit its expression. Thus, the Wnt pathway genes whose transcription is activated by the beta-catenin may not be expressed in our HCC 1937 samples, leading to an observed downregulation of the pathways in these cells. This further accentuates the heterogeny even within triple-negative breast cancer subtypes as we demonstrate diversity in beta-catenin expression. The results of this paper demonstrate a use for integrating multiple omic data types together for the goal of investigating the interconnected nature of biological data that a single omic data type may not be able to demonstrate. In the future, we hope to develop our own machine learning algorithm that does not only rely on simple correlated features, but can use multiple omic data input and provide a comprehensive report with a ranked list on the most important features and pathways within a dataset. Finally, I want to thank everyone for making this research possible, and I hope everyone has enjoyed my talk today.